am Dr. Anna Bessoren, and it is my pleasure to have you all here this evening. I had the opportunity to work with all of these students since spring of this year, while they identified an area of interest and someone in the community who would mentor them. Several of those mentors are here tonight, and I will point them out and thank them later after you hear about the wonderful work they helped facilitate this summer. After their summer research, students came back to St. Mary's this fall and have been working very diligently on putting together research papers and also the presentations that you are about to see. I have been very impressed by the work that all 5 of these HIRS students and then 1 additional student who had the opportunity to do work at St. Jude were able to do, and the dedication that they've shown and the effort that they've put in. So since you did not come here to hear me, I would like to go ahead and get us started and introduce our first speaker.
Hi everyone, my name is Alexandra Lafferty, and this summer I had the opportunity to work with Dr. Kieran Kahn at Rhodes College and an undergrad student, Abby Holtzquist. For my HIRS project, I wanted to research how group dynamics affect child learning. So to start off, Summer Success is a kindergarten readiness program implemented in the summer of 2019 in Memphis. It went over math, behavioral, motor, language, and literacy skills. And part of the program was a storytelling circle, where 4 to 5 children were taken out of their classroom and put into a storytelling circle. This happened 3 times a week for 4 weeks, so 12 sessions total. And this happened over the course of 2 months. And during these 2 months, they were split into 2 different groups. So that would be June Circle 1 and 2 and July Circle 1 and 2. So each child was given a pre and a post test to determine what level of preparedness they were for kindergarten. So to start off, the pre and the post tests were the same story, and so each child was told to give a true story about themselves in every session. And so in the pre-test and the post-test, it was tell me a true story about a time you felt sad or frustrated, and some examples from this graph you see are from session 4, tell me a true story about someone made you about a time someone made you cry, and tell me a true story about a time when you fixed something. And so there were 4 to 5 children, as you can see here, per 2 facilitators. And so every session was recorded to later be analyzed, which is what I did. And so each story was then coded for level of character representation sophistication. And so the simplistic levels are actor, which is levels 1 through 3, which just means their story had the least amount of action or emotion in their story. Agent is the middle level, levels 4 through 6. So that means that they showed some levels of emotion and action, but not at the highest level. And the highest levels are person, which are 7 and 8, which means that they showed a lot high levels of emotion and action. So from those, we discovered that in the later sessions, children tended to be at a higher level, which are agent and person, and the stories were split up from self if the story was about themselves, or if the story was about another person, but it's still a true story. And so as you can see, the children tended to be at an agent level or person level rather than an actor level. This is another way of showing how in the later sessions, the children tended to be at a higher level, which is levels 1 through 8. And if there's a dot missing, that just means there was data missing. And then the children were split up by age, 4 and 5-year-olds. And as you can see, the children tended to be at a agent or person range the older the child was. So for my project, we went over character representation sophistication. And so we discovered that 10 out of the 20 children made clear gains in this, and 8 out of the 10 children came from June Circle 2 and July Circle 2. So what I did over the summer is I analyzed about 150 stories, which were recorded, and we looked for possible factors impacting their performance. So I looked for eye contact, movement, talking, laughing, what we called influencers, which are people who positively or negatively affect the group. We looked for friendships, downtime, and playtime. So what we discovered is that there were higher levels of frequency of collective laughter and talking in June Circle 2 and July Circle 2. And there was a higher number of influencers in these two circles. So the relevance of this is that talking is important for development. Narrative learning creates a positive and learning environment, and storytelling is important for language development. Preschools would benefit from this by incorporating more verbal collaborative activities, which would better prepare students for kindergarten. I would like to thank Dr. Kieran Kahn, Associate Professor of Psychology, Abby Holtzquist, Dr. Annabeth Soren, and my fellow HIRS classmates.
Hi, I'm Charlie LeMountain, and this summer I had the opportunity to study the effects of parasitism on prothonotary warbler parental behaviors. I worked with Dr. Keith Bowers and graduate students at the University of Memphis to study this. So first, prothonotary warblers are a species of songbird. They're pretty small and bright yellow. They're quite beautiful birds, in my opinion. I spent a lot of time looking at them this summer. In the wild, the development of the range of these birds is between North and South America because they are a migratory species of bird. They spend their summers in North America where they raise their nests and their nestlings, and then they migrate down Central America and South America for the winter. We studied this species of bird at the Neiman Biological Station, which is at Shelby Farms. So the development of the nestlings takes place over a really rapid period of time. The difference between the picture on the left and the picture on the right is about 11 to 14 days. So the development of these nestlings happens really, really quickly. And because of this, we wanted to understand exactly how the parental decisions that the adult birds makes affects the success of their offspring. There are two types of parasites that affect wild bird populations, including prothonotary warblers. The first is blowflies. Blowflies are an ectoparasite, meaning they lay their eggs in the nests of wild birds, and they also lay eggs on the skin of animals. The eggs then hatch, and the larvae of blowflies feed on the blood of the nestlings or of the animal that they are laid on. The other species that we studied is the brown-headed cowbird. Brown-headed cowbirds are a nest parasite, so they lay their eggs in other birds' nests and then leave their eggs and their nestlings to be raised by the host nest. So in the picture on the right, you can see the difference between a prothonotary warbler nestling and a cowbird nestling. The cowbird nestling is noticeably larger, and they are also more aggressive with their begging behaviors, which affects the success of the host nestlings. There are three hypotheses for how parents choose to feed their nestlings based on how to maximize success of the nest. The first is signal of need. So this hypothesis says that parents choose to feed the weaker nestlings in order to maximize the success of all the nestlings overall. So if you feed the weaker and lighter nestlings, then all of them are more likely to survive. Signal of quality says that the parents are more likely to feed the stronger nestlings to ensure that at least a couple of the strongest nestlings survive from the nest. And the last one is first come, first serve. This hypothesis simply says that the birds choose to feed the first baby that opens their mouth. So whichever nestling begs first will receive the food. We conducted our study at Shelby Forest. We set up nest boxes all throughout the field site and use these as nests, or as birds continue to nest in them, to study their behaviors. So in order to keep track of which birds laid eggs in which nests, we would band them. If you can see that in this picture, this male prothonotary warbler has 4 different colored bands on his legs, and this helps us keep track of which bird is which. The lids of these boxes are also removable in order to allow us access to the nestlings and to the eggs, so that we can conduct our experiments and our research. So for my experiment, I wanted to be able to control for the presence of parasites in the nest so that I could further analyze how the decisions of the parents change based on the presence of parasites. So for blowflies, I did heat treatments, which is where I would take a heat gun, as you can see in the picture, and I would heat first the inside of the nest cup. I would move on to the outside of the nest, and then I'd flip the nest over, do the same thing, and also heat treat the inside of the box. This would ensure that the blowfly larvae were not present in the nest anymore. The nestlings would be removed for this process and weighed to keep track of growth. A control treatment for blowfly parasites was we would remove the nest to simulate the amount of time that the nest would be out for the heat treatment, just so that there was more control across both experiments. For cowbirds, the presence or the lack of the presence of the nestling was how we determined whether the nest was parasitized or not. In order to keep track of the decision-making of the birds, we used GoPro cameras inside of the nest boxes. We would record for about an hour to 2 hours for each nest on 2 separate days, on day 4 of development and day 7 of development. So in the picture on the left, you can see that both the male and the female parents have arrived to the nest, and they are both separately feeding different nestlings. Each of these is a nestling. You can see this one has a silver band. They were all banded. And they also have these small white dots on their heads. This allowed us, based on the positioning of the dot on their head, it allowed us to tell apart the different nestlings so we could keep track of who was begging and the size of the nestling and how strong they were, etc. In the picture on the right, you can see that the female has come to the nest to brood. She's keeping the nestlings warm in order to allow for them to put their energy into developing and not trying to keep themselves warm. So after collecting all of our data, in the video footage, what we found was that in control nests, the ones that were parasitized, the nestlings were begging more. And then in the unparasitized nests, the nestlings were begging less. So this shows that when nestlings are having to expend more energy on being parasitized, they also need to beg more to receive more food. From the parental aspect, we saw that when the nest was parasitized, this is the difference between the weight of the nestling that is being examined and the mean weight of the nestlings in that specific nest. So for negative numbers, the nestling being looked at is smaller than average, and for the positive numbers, the nestling is larger than average. So this allowed us to see the changes in parental feeding decisions based on weight. So in a parasitized nest, the parents would largely choose the stronger and larger nestlings, and in an unparasitized nest, they would feed the weaker and smaller nestlings. So what this shows us is that in parasitized nests, the parents would exhibit the signal of quality decision-making, showing they would feed the stronger and larger nestlings to ensure that at least some of their genetics were passed on to their offspring. In an unparasitized nest, the parents would exhibit signal of need decision-making. So they would feed the weaker and smaller babies in order to maximize the success of the entire nest, because they had the time and energy to put into all nestlings rather than battling against parasites. I would like to thank all of the people I got to work with for this project, especially Dr. Keith Bowers and the graduate students at U of M, as well as Dr. Annabeth Soren and my fellow HIRS classmates. This was an incredible experience.
Hello, my name is Rachel Larkins, and over the summer I was able to look at allelopathic impacts on tree seedlings and germination and growth, and that's a lot of big words, which essentially means how species interact in their biochemical environments, and specifically non-native versus native species. So over the summer, I was able to work with Overton Park Conservancy with the mission to better improve and protect the forest just for the overall greater Memphis community. So as a little bit of a background, native versus non-native species. So what makes a tree native is when it has co-evolved with another species. So they've had an equal opportunity to adapt to one another and create equal biochemical environments. A non-native tree is when you introduce a tree which has not had the same chance to evolve So then it can have advantages over the trees that it has been introduced to. This can have a lot of different risks to the ecosystem, as a lot of non-native species can be invasive and take over. This can be damaging to the regeneration processes of the native species, such as seed dispersal and other ways in which they spread. Humans have the greatest impact on introducing these non-native species. A lot of people don't realize in their landscaping and in their personal gardens, they introduce a lot of species which then spread to their surrounding forests. A great impactor on this one is the zoo for Overton Park, because it's in such a close proximity. And a lot of the trees and species which they use for their exhibits are non-native and then essentially migrate into Overton Park. So what is allelopathy? Allelopathy is when biochemicals are released into the environment, and this can happen in a variety of different ways, such as through the leaves decomposing, through the roots, and even just through the air and being in a similar environment. For our experiment, we looked at when the donor plant is a non-native species and when the receiver plant is a native species, and so the donor plant would give off the chemicals impacting the native species. So coevolution is when these two species are native, they have co-evolved, and they have the same opportunities to affect one another, so they're less harmful, and they've adapted their allelopathic relationships. So when the donor plant is non-native, it can have a negative impact by having advantages such as like creating a favorable environment for its own seedlings. For this, it can look like the leaves assisting their own germinants or just helping their growth. So the specific non-native tree that we looked at over the summer was the cherry laurel. It's a non-native. It's native to the Mediterranean region. It's very fast spreading, and it is prevalent in Overton Park. So we completed a survey that was also completed 32 years prior, and the cherry laurel had the most increase comparable to other species, and it is an understory tree. That's the reason why we chose to pick this specific tree to look at. A lot of the overstory trees that we found in this survey were decreasing in growth, so that's why we had a suspicion that this could have an impact. So the treatment that we created is a concentrate of the biochemicals. So we extracted the biochemicals from both the roots and the leaves of the cherry laurel. So the non-native plants can inhibit the growth of the native plants. The specific overstory trees which we looked at were the tulip poplars, northern red oaks, and sugar maples. The reason why we chose these is because all three of these had decreased from the previous survey, and they all have a variety of different qualities. So if it does impact a certain quality of one of these trees, it can change their growth patterns. An example of this is the early successional, mid-successional, or late successional. This is important because in the forest there's a lot of competition, and so the first one to the light will be the most successful plant. And so if it can be inhibiting their growth and slowing that down, the cherry laurel can take great advantage over that. So for our research study, we put 5 seeds in each Petri dish and then randomly assigned a treatment. The treatments were A, B, or C. A was 100% cherry laurel root or leaf concentrate, and then the B treatment was 50% distilled water, 50% cherry laurel concentrate, and then the C was just 100% distilled water. And then they were, once they germinated, which we determined to be having a root radical of 2 millimeters or greater, then they were moved into a tray here in the cells, and then we kept track of the dates in which they germinated. They were then moved into the greenhouse, and they were kept at a consistent light, heat, and humidity. There they were able to grow for 10 weeks, and we watered them 3 times per week. So the measurements we collected from this were biomass, which is essentially the mass of the tree. We dehydrated them in the dryer oven and basically took out all the water and then weighed each of the trees. We separated them into above and below ground samples because there's been studies shown that when a tree is under stress, it can cause the roots to grow more in order to save itself and the stems to grow less. So we're trying to look at that and the ratios between them. We also looked at the stem height and stem width and leaf length and leaf width. For this measurements, we specifically looked at the northern red oaks because they were the easiest to measure too. So the average diameter and average height of the northern red oaks didn't show that there was any statistical differences between the 3 treatments. And then also the average survival and both leaf length and width didn't show there was really a statistical difference between the 3 treatments overall. In conclusion, there's still current measurements being taken. This could be just the northern red oaks is what we collected our data on. So if in future studies, we could look further at the sugar maples and the tulip poplars. Future studies could also be relating to the field. We also could look at the seedlings, what makes them, or predation, like the cherry laurel could have properties which make it less likely to be eaten by predators or scavenged in the forest. So this is really important to the tree life cycle because trees do die off. As they die, they need to be replenished in order to maintain biodiversity. For forest management improvement, this is really important to reduce like expensive projects which would rip out all the cherry laurels in the whole Overton forest. So if they're not making an impact, we can know better where to spend our money to help the trees. I'd like to acknowledge Overton Park Conservancy and my mentor, Eric Bridges, our supervisor, Dr. Annabeth Soren, and my fellow HIRS students.
Hi, my name is Shifa Panjwani, and over the summer, I worked with the Community Legal Center to research the lack of county attorneys for the elderly in Tennessee. The problem is this increase in elder abuse, as the data shows. So especially over the past 3 years, in 2019, 2020, and 2021, there has been a steady increase in elder abuse, rising from 146 cases to 207 cases. The CDC defines elder abuse as an intentional act to harm an elderly person, usually as a result of neglect or isolation. There are 3 main kinds of abuse: physical, sexual, emotional, or psychological. Physical refers to any violent activity towards the elderly, sexual refers to unconsented activity with the elderly, and emotional refers to anything that becomes mentally negative, mentally challenging to the elderly. Elder abuse happens when an elderly person is either isolated or they lack certain abilities, or both. By lack of certain abilities, it could be that they are mentally less aware than before, or they're lacking physical strength. Conservators are needed when the elderly are either being abused or when their capacities are lost, even in the absence of abuse. A conservator is someone who acts as a guardian on behalf of the elderly when they are unable to make their own decisions for themselves. Pretty much anyone who is interested in the best future of the elderly can be a conservator, but some examples include the child of a conservator, their spouse, a close relative, or just really anyone else. Even a state guardian could be a conservator. The reason we should care about this problem is because it affects our lives and possibly our families' futures, but also it just affects us as a community, especially as baby boomers begin to reach the age where they will possibly need conservators. When an elderly person receives a conservator, some of their personal rights are granted to the conservator and therefore taken from them. There's something called the least restrictive alternative standard in Tennessee and in other states, and that standard refers to the fact that the elderly people will have to give away some of their rights to the conservator, but only the absolutely necessary rights will be given to the conservator, and the rest of the rights will remain with the elderly if possible. So the rights are assigned based on the functionality and decision-making of the elderly. The functionality of the elderly are examined based on if they have communication abilities, such as are they able to describe their wants, needs, and desires, and are they able to perform basic daily activities. Also, their decision-making is analyzed while assigning rights. So are they able to make major decisions such as healthcare decisions and property decisions, but also are they able to decide what daily chores they will do, such as will they be able to clean the house that day. There are three kinds of conservatorships: full, partial, and state of guardian. So a full conservatorship is when a conservator handles the economic and healthcare decisions on behalf of the elderly. A partial conservator handles only the economic or healthcare decisions on behalf of the elderly, and a state guardian has full conservatorship over the elderly, but they are not a personal guardian, they are by the state. The legislation of Kentucky and Tennessee as far as receiving a conservatorship are very similar. So the first process of petitioning, which is done by someone who is interested in the best future of the elderly, and they notice that the elderly person needs a conservator to act on their behalf, they'll file an initial petition to the court, and then an attorney will provide evidence to the judge to actually create that petition. So in the petition, there's the witness testimony, which is given by someone who notices this need for a guardian in the life of the elderly. And then a medical record is given by a doctor to demonstrate the physical, mental, or both need of the elderly to need a conservator. And the desire of the elderly in relation to what conservator will be assigned to them are also considered and put in this petition. The judge will then take that petition and make a decision of who they think will be the best conservator on behalf of the elderly. The only real difference between a conservatorship in Kentucky and Tennessee is the actual attorney representing the elderly client. So in Tennessee, we require a private attorney or a low-cost attorney at an organization such as the Community Legal Center or other similar nonprofit organizations. And Kentucky has a free county attorney, and that is an attorney given by the state to those individuals who cannot afford to hire an attorney for themselves. So I researched the Kentucky revised statutes, which talk about the roles of the county attorney and what exactly their position does. In Tennessee, because a private attorney is required as opposed to a county attorney, there is an economic disparity seen. Those who have money to hire an attorney are able to receive conservatorships, while those who do not have the financial funds cannot hire an attorney to then receive a conservatorship. So a lot of my research was going through a lot of lengthy legal documents to understand the roles of the county attorney, which were defined in the legislation of Kentucky, the Kentucky revised statutes. So the county attorney needs to defend civil action and do what's in the best interest of their constituents. In my case of research, that was the elderly, and they do this by providing evidence in order to assist those who are at risk of exploitation in our community, which in this case is the elderly. So their main job is to assign a responsible conservator to the elderly, but also if a conservator becomes abusive, they are able to appeal to remove the conservator. And the attorney does this by contacting the clerk, either by calling them or writing a letter to make sure that the judge can set a date to look into the removal of a conservator who becomes abusive. Conservators generally become abusive economically, and that's by stealing money from the elderly without their knowledge. So through these roles of the county attorney, their goal is to assign this responsible conservator, but also to make sure that the elderly are protected. Some of my research also talked about the salaries in terms of the county attorney. So the county attorney is paid by taxes because they are a government paid position, and they have a 4-year long term. So every 4 years, the people in the county will vote on the county attorney. Tennessee would definitely benefit from this county attorney, which is currently a position in Kentucky, because the biggest economic burden of receiving a conservator, which is the actual cost of the attorney, would be completely eliminated. Also, there would be ease of access when trying to get an attorney to represent the case of the elderly, because there would only be one option, which is the county attorney, so it would be much easier to access. Also, there would be further awareness raised on the need of a conservator, as more people would begin to receive conservators with the presence of a county attorney. There are some constraints to creating a county attorney in Tennessee specifically. So there's first the fiscal constraints with tax distribution. So right now there is not a tax allotment for the position of county attorney in Tennessee, but the reason for this is that there is a lack of awareness among the general population and especially among attorneys on the actual position of county attorney. So as that awareness is further raised, hopefully a tax allotment can be created. This image right here shows where in all the 50 states where different taxes are collected, and those taxes are then distributed to different services, organizations, and positions in each county. So there is an increase in elder abuse in Tennessee, and even for the elderly who are not abused, they may lack some physical or mental abilities which make them need a conservator to act on their behalf. Tennessee should definitely add the position of county attorneys to help the elderly who cannot afford the cost of an attorney but still need a conservator to act on their behalf. As a long-term goal, I hope that the tax allotment in Tennessee in each county is reconsidered, and they can do this by first increasing awareness amongst the general population and especially among attorneys. I want to thank Karen Henson for helping me with my research over the summer at the Community Legal Center, and I want to thank Dr. Soren for her help with the research paper and this presentation.
Oh.
Whoops.
Sorry, guys.
Maybe not. This, that. Oh, that product deal. Beautiful.
Sorry. So after that, I'm Daryl Soot, and I will be talking about education and workforce training for people with disabilities, which I could study by working with an organization in Spain this summer. So what does it mean to have a disability? A disability is a physical, mental, cognitive, or developmental condition that impairs, interferes, or limits a person's ability to do certain tasks or participate in certain daily interactions. And so the Individuals with Disabilities Education Act, also known as IDEA, identifies 14 different types of disability and gives us a rough breakdown percentage of the students with disabilities. The main ones being specific learning disability, like dyslexia, dyscalculia, and dysgraphia, speech and language impairments, like stuttering, other health impairments, like ADHD, and autism spectrum disorder. The least common ones being deaf and blindness, deafness, visual impairment, and traumatic brain injury, with less than 1%. It is said that 1 in 4 US adults lives with a disability, which means that it's a pretty common thing today. So why is there still a negative social construct regarding disabilities? I found it interesting how social attitudes towards disability have been artistically represented throughout the years. Disabilities started being portrayed in art during the Middle Ages. Like here we can see Caritas by Bruegel the Elder. It is a group of homeless people. Some of them have a disability, mainly physical because it was the one they could see at the time. And the Latin quote underneath is telling the audience to help them. Then we have the beggar with the wooden leg in the Renaissance. The Renaissance starts having an increase in interest in the characteristics of the disabilities. So here, there is a shadow cast over the wooden leg, which is triggering the audience to shame it. There's almost a sense of disdain over it. And then today's stigma is really based on the stereotype developed during the 19th century, which was influenced by the Industrial Revolution. In Maniac in Chains, we can see the person chained to the wall, and he has almost this beast-like appearance. And so, as we can see, even though throughout the past few years, we've been trying to destigmatize the prejudice over disabilities, it's still pretty common. But there has been an increase in disabilities among our population. The reason for that increase is uncertain, whether it is for an actual increase in disabilities among our population or for a more accurate diagnostication. We don't know. Maybe now we understand disabilities better, which is making us see that they're more common than what we think. However, the lack of education and workforce training for them is then represented in the employment rate. People with disabilities have a 40% less rate than people without disabilities in employment. An interesting fact, though, is that the pandemic actually increased the employment rate for people with disabilities because of working at home. Maybe someone had a physical limitation, and so going to the workforce made them not be as productive as they could, or maybe it was sensory issues, and it's the same. Being in that workforce environment wasn't the most beneficial for them. However, there is a big change, which is then reflected in the poverty rate. People with disabilities have a 15% more chance of ending in poverty than people without. And so I worked with Amica, which is a nonprofit organization that started in 1984 in the north of Spain. And it works on discovering each person's capacities and giving them opportunities involving society. The interesting thing about Amica is that it works with every single type of disability.
And so a person with a disability will go to Amica with their family and they'll have a meeting. And so Amica will propose a personalized program for them depending on their capacities and their needs and wants. And so it can range from maybe speech therapy for a kid who has a stutter who will go after school to maybe an older person whose goals in life are not being completely independent and getting a job, so they will go to adult daycare. They do all of this through personalized learning systems. Personalized learning is the creation or development of educational models made to fit the students as needed. So it's more student-focused, it allows for more inclusion, and it gives more support systems. Amica specifically uses the causal agency theory, which was developed by Michael Waymeyer. Basically, they teach them what they need to learn, as well as helping them develop their capacity and creating a safe environment and giving them the experience needed to increase their opportunities. That, with Amica's support, will develop their autonomy, empowerment, self-regulation, and self-realization, which then will simultaneously increase their self-determination. This, as everything else, depends on each person's capacities. Maybe for a person in the adult daycare, being self-determined means picking the puzzle that they're going to do that day, or what colored paper they're going to draw on, or what shirt they're going to wear. Maybe for someone in a workforce training, that means getting to be completely independent and getting to get a job and choosing what they're going to do. So the counselors will assess the students on a monthly basis using a numeric scale and answering these 6 parts: autonomy, auto-initiation, auto-direction, self-regulation, auto-concept, and empowerment, answering several questions in there. Do they have personal hygiene? Can they use public transport and move around their community? Are they aware of their limitations, and do they know how to work past them? And so for the people who are in the workforce training, Amica then has several work centers where they can work at. They have a few laundromats across of the north of Spain, as well as clothing manufacturing. An interesting thing is that Amica manufactures and cleans the clothing used in the north of Spain healthcare system, so the hospitals. They also have recuperation and recycling centers, which is the picture we can see here. That is one of the centers in Santander. And environmental cleanup. This year, they proposed cleaning parts of Spain of an invasive species. I worked most of the time with a workforce training for people between the ages of 16 and 26 for them to work in the laundromat. So they would spend half of the day in the laundromat getting used to how the machines worked and what they had to do, and the other half working on that self-determination. Amica uses support system, which means that they will assess the students, in this case for the workforce training, on how the machines work and how to use them, and they will tell them what they're going to do each day. But then if they were seeing that someone maybe was losing speed and was going a little slower, instead of telling them, you're doing this wrong, a counselor, I did this a couple of times, would go next to them and start doing the same work as them, but at a faster rate. So they would see it and copy it. This, working with this for several weeks, allowed me to see how it improves their quality of life, and how because they offer different programs and work with all types of disabilities, they touch on a much bigger part of the population. They're also socially active and are triggering social change. So destigmatizing that negative stereotype over disabilities. And they offer the people and the participants many opportunities for them to thrive in that environment. So really, Amica is doing a great job, and they're using their system really works from what I've seen, and is maybe something that we should start copying a little more. I would like to acknowledge Amica, the organization as a whole, especially Noelia Ojea for being my mentor and teaching me all of this, Noemi, Esther, and Gonzalo for facilitating my entrance in Amica and showing me 2 different centers, Ms. Priliman for helping me with the art historical representation of disabilities, Ms. Akers for assessing me in the research, and Dr. Sorin for helping me on the presentation and the research paper. Thank you.
Oh, it's already there. Hi, I'm Sophia, and this summer I studied the impact of genetic mutations on pediatric medulloblastoma. I had the opportunity to work under Dr. Hong Lin in the Developmental Neurobiology Department at St. Jude Children's Research Hospital in the Northcott Lab. I participated in the high school and college research immersion program for 8 weeks. So the most basic definition of cancer is uncontrolled cell growth. This is due to increased proliferation and lack of differentiation, which means that cells are over-multiplying and over-dividing, but not taking on a specified function within the brain or the body. I studied the brain. We studied a specific subtype of cancer found in the cerebellum, which has the arrow pointing to it here. The cerebellum deals with motor function and coordination. So when its development is disrupted, it's really easy to tell the signs, especially in the earliest years of life when children are learning how to walk and move about the world. We particularly studied sonic hedgehog medulloblastoma, which is a specific type of medulloblastoma. Here's a photo of a cerebellum under a microscope. As you can see, during normal development, there's 10 lobes, and these neurons follow a very specific developmental period in one of these cross sections here. So if you take a closer look, you'll see that neurons originate in the external granule layer, in this layer right here, the external granule layer mitotic layer, which is where they undergo mitosis and divide. After the neurons proliferate, they then move to the EGLD, the external granule layer differentiating layer, where they take on a more specified function in the brain and they mature fully. And during normal development, they'll form these nice organized lines of neurons, which then migrate to the inside of the cerebellum, and that's where we start to see real growth of the brain. We studied a specific protein found in the cerebellum called the elongator complex protein 1. We've seen a lot of data indicating that when the gene that produces this protein loses its function, that children are much more likely to develop this certain subtype of cancer. So we sought to explain why. We hypothesized that the mutation of ELP1, the loss of function of ELP1, deregulated the process under which granule neuron progenitor cells differentiate and proliferate, meaning that the entire process of development in the cerebellum is disrupted. To study this, me and my partner followed a very specific methodology. We began by isolating both wild type and mutant samples, which means samples with the function of the ELP gene and samples that have lost the function of the ELP gene. And then we embedded those samples in paraffin wax. Then we used a microtome to cut ultra-thin slices of the samples, and then we performed immunostaining, which allowed us to compare the relative protein levels at relevant points during cerebellum development. Then we mounted our samples and took light field microscopic images of them. So here's a summary of some of the data that we collected. As you can see across the top, we have our 3 stains, HE staining, P27 staining, and Ki67 staining. And we have 3 separate time points, P7, P10, and P14, with both wild type and mutant samples in both low magnification and high magnification. So HE staining stains for the basic morphology of structures. It stains both the cytoplasm and the nucleus of cells, and it gave us a good benchmark to base everything else off of. P27 stains for the proteins that are secreted while neurons take on a more specified function in the brain. So while differentiation is occurring, they secrete proteins, and this stain latches onto them, and it allows us to view it under a microscope. And then finally, we stained using Ki67, which stains for the proteins secreted while the neurons are dividing and undergoing mitosis. And here's an example of all of those low magnification images of the cerebellums at P10, which is a specific time point, using the 3 stainings, both wild type and mutant. And if we take a closer look, we'll start to see a very significant difference between the wild type and the mutant samples. Using the proliferation marker, we can see that the size of the proliferating layer has nearly doubled in size. And also, the intensity of the dye is much darker in the mutant sample. As you can see, this color here versus the color of the wild type sample. So when ELP1 loses its function, more proliferation is happening at a much faster and larger rate. However, if we look at the differentiation marker, we can see that differentiating has decreased in both size and intensity, meaning that these cells are not taking on specified functions within the brain at the rate in which they should, at the rate in which they do in the wild type samples during normal development. Additionally, if you think back to that first
image I showed you of the cross-section when the neurons are developing, they're supposed to form these nice, clean, organized lines. But what we see in the mutant sample is that the neurons are all jumbled together. And this is just another indicator that the development of the cerebellum has been completely disrupted. And at later time points, using the differentiation marker, we can see a complete lack of differentiation at all. In these red circles, we can see that no dye has appeared at all, which means that no differentiation is occurring, and cells aren't taking on a specified function whatsoever. So our results, therefore, implicated the role of the ELP1 mutation in the tumorigenesis of medulloblastoma. Our study explained the reasoning behind why the loss of function of ELP1 leads to an increased likelihood that a child would develop a specific type of medulloblastoma. And while we're still working to quantify our results, since I've shown you visual data, the difference is apparent nonetheless. In the future, complementary studies could look at different genes and compare different relative protein levels. For example, one gene is the P53 gene, which is a tumor suppressor gene. So to observe its lack of function could be very interesting as well. You could also compare sonic hedgehog levels, which is another nickname for a protein found in this type of the brain, in this area of the brain. And then within the larger preclinical trial process, this is us providing the understanding behind one pathway that this specific subtype of cancer takes. This allows us to engineer more targeted treatments for the patients that come into St. Jude every day with the lowest toxicity. I'd like to thank Dr. Paul Northcott and Dr. Hong Lin, as well as the rest of the members of the Northcott Lab and my partner, Anthony Bruce. I'd also like to thank the program managers at St. Jude, Dr. Annabeth Soren, and my fellow HIRS classmates. Thank you.
Okay, so I hope you got a glimpse at what these students have been working on and dedicated their summers to. I would like to thank the mentors that are here, Dr. Bowers, Dr. Khan, Dr. Lin is back maybe, yes? So I'm very glad that Memphis is such a welcoming community for our students. And every year, there are always community members who are willing to take on St. Mary's students and welcome them into their labs or into their nonprofits and give them a real immersion in what the type of work they do involves. And that's a really unique experience for high school students. And I'm always very appreciative of what those mentors do. I feel there are probably some that are online that I want to thank as well. What I would like to do now is open things up to the audience, and all of the girls will stand. If you have any questions of them or comments about what they did this summer, we welcome your thoughts. And then once they are finished in their very tight little lineup there, please join us in